Hello students, good morning. Myself Jayashree Vilas Rundor from Department of Chemistry. uh, KANMS Art Commerce Science College, Satana. Today we discuss about topic nucleic acids from medicinal chemistry. Nucleic acids. Nucleic acids are long chain polymeric molecules. That is, in nucleic acid, the monomer or repeating unit is a nucleotides, and hence it also referred as polynucleotides. It also defined as an organic molecule present in living cells, especially DNA and RNA. It play a key factor in transferring the genetic information from one generation to next generation. DNA is a complex of pentose sugar, phosphoric acid and some cyclic bases containing nitrogen. The sugar present in a DNA is deoxyribose, that is beta D to deoxyribose. The cyclic bases that have nitrogen in them are adenine, guanine, cytosine and thiamine. These bases and their arrangement in a molecule of DNA play an important role to storage the information or transfer the information from one generation to next generation. DNA has a double strand helix structure, that double helix structure in which strands are complementary to each other. While in RNA is a ribose nucleic acid. It also composed of a phosphoric acid, pentose sugar and some cyclic bases containing a nitrogen. In RNA, containing a beta D ribose sugar. In RNA, the bases are adenine, guanine, cytosine and uracil present. In RNA, there is a fourth base is different from that of a DNA. It also consists a single strand with some time of fold back. There are three types of RNA. First is a messenger RNA called as mRNA, ribosomal RNA, that is rRNA, transfer RNA, that is the tRNA. There is a functions of nucleotides. Nucleotides are responsible for the transmission of information or inherent characters from parent to offspring, responsible for the synthesis of protein in our body. DNA fingerprinting is also used by forensic experts to determine the paternity and also the use for identification of chemicals. Next, the nucleotides. We already seen that nucleotides are the repeating units in nucleic acids. There are three types of nucleotides. First is the purines and pyrimidines. Purines, there are three parts in purines. First is a nitrogen base ring, that is base, then pentose sugar. In DNA, the pentose sugar present deoxyribose, while in RNA, the pentose sugar present is ribose sugar. While third is a phosphate group. These three parts are form the nucleotides. The next is types of nucleotides. There are two types of nucleotides, purines and pyrimidines. Purines are the examples of purines are adenine and guanine, while pyrimidines are cytosine, thiamine, uracil. In purine are one of the two chemical substances that cell used to create the constituent part of DNA and RNA. It also heterocyclic aromatic chemical composed of two fused rings. Okay, it dissolve in waters. The body convert them into uric acid, which is excreted in urine. While pyrimidines are also one of two kind of heterocyclic nitrogen base ring which used to create the constituent part of DNA and RNA. That's called a pyrimidines. In pyrimidines, there is presence of single heterocyclic nitrogen bases, while in purines, there is presence of a heterocyclic aromatic rings of two fuse rings. Nucleic polymer. We already see nucleic acids are a polymeric chain. Polymers means a monomer that is continuously repeated. In nucleic acid, nucleotides are repeated, so they are called as monomer in nucleic acid. Next is hydrogen bond DNA molecule. Okay, hydrogen bond is very essential for DNA molecule. Hydrogen bond are extremely important because they are the responsible for structure of DNA and its property. It is responsible for connection between the two strands of DNA between nucleotide base pairs at a middle. Without hydrogen bond, DNA will not have exist as different structures. Okay? Hydrogen bond is very essential for DNA. There, copying DNA, there is a replications of DNA. Two strands of DNA helix are complementary. Have one can build the other and have one can rebuild the whole. When does a cell copy DNA? Cell reproduction, mitosis, etc. Rosalind Franklin. Rosalind Franklin was a British chemist and X-ray crystallographer whose work was central to understanding the molecular structure of DNA and RNA. There is number of hydrogen bond ratio in DNA molecule, 2 hydrogen bond versus 3 hydrogen bonds. Okay, we see that thymine and adenine have a 2 hydrogen bonds, while between a cytosine and guanine there is a 3 hydrogen bonds. Thank you.